name is Michelle Morand, and I'm here with the cancer guy, Alexander Rolland. And today, Alex wanted to come here and talk with you about a tool that he has helped develop this last couple of years that is just a real game changer for cancer patients and for their treatment teams in terms of giving them far more options to work with for cancer treatments and far better outcomes. So Alex, I'm going to turn it over to you. You've got a little bit of a slideshow that you want to share with us, I think. Please share with us.
Okay. Well, before I get started on that, I'd just like to tell you a bit about this technology. So it's many years, we've been trying to introduce new technologies that are used in the development of oncology drugs. We're trying to bring them into the clinical practice. It stands to reason that these tools have been used to create these drugs, then they should be used to better stratify which patients they're going to help. So a little primer beforehand. A lot of precision oncology has been based on what we call tumor DNA sequencing, and I'm sure everyone's well aware of that. You isolate the tumor cells from a biopsy, and then you look at a panel of genes and try to find what cancer-causing mutations are there, and then have targeted therapies target those specific mutations. And there's many examples of this. Most cancers have different targeted therapies, that you can use tumor DNA sequencing to determine which are best for it. However, cancers also use another way of growing and metastasizing, and this has kind of been the holy grail. And understanding this really has been the crux of cancer treatment in precision oncology. So in short, this is called epigenetics. And what epigenetics refers to in a generalized term is the alternate turning on and off of different genes in order to create different tissue subtypes. And so, as you know, in theory, every cell in our body has exactly the same genome. If I were to take a sample of a skin cell and a sample of, let's say a liver cell or something, and sequence them both, they're going to have basically the same genetics. The genes and the alterations are going to be somewhat similar. And, that we can use that to identify who a person is, based on that genetics. However, our body is composed of a myriad of different tissue types and different cell types. And so a lot of people wonder, well, how does that same set of genes create all of these different tissue types? Well, the answer is epigenetics. And in the easiest term, what epigenetics refers to is turning off and on different genes to create these different tissues. So we have a massive amount of genes and alternate splicing and different tools for creating proteins. And the vast majority of it is designed or has evolved over evolution to control how long a gene is turned on for and how long it's turned off for, and when it's turned on, in order to create all of the different tissues. So in theory, we have the genes that could potentially grow a tail. It's just that they're not turned on in humans anymore. And so what cancers do is they actually turn on genes and turn off genes that are supposed to be turned off and on. And understanding which genes are turned on and which genes are turned off is really important in determining what treatments are best for a patient. And as you know, most drugs, including chemotherapy drugs, will actually affect certain genes. And whether those genes are turned on or off can tell us whether certain drugs, including chemotherapy drugs, will work, and as well as radiation therapy, and of course a myriad of targeted therapies. So I'll talk a bit about this tool. First, I'm going to open up my slide here. So this tool is called Tumor RNA expression testing. And basically what RNA expression testing is RNA is an intermediate between the DNA template that encodes for the gene and the final product, which is referred to as a protein or an enzyme, whatever that gene creates. And so basically what happens here is the DNA will create a certain amount of RNA molecules, and those RNA molecules will then get converted into proteins. And so the length and time and amount that a gene is turned on can be determined by measuring the RNA for that gene. And so for many years, RNA expression testing has just been very expensive and not really usable. Mostly because the RNA technology has been based on a tool called RNA sequencing. And RNA sequencing basically was designed to look at mutations in DNA. They applied it to RNA, but really it's designed to look at mutations and detect mutations in RNA. It's not really just designed to determine how much of that RNA molecule is expressed or not. In other words, how much of that gene product is being produced. So about 2 years ago, a company created a new tool that is designed specifically for RNA expression testing, and it's using next generation sequencing, which is the latest technology, but it's an automated process. And right now we're looking at a massive panel of genes. So what this does is it allows us to isolate the RNA from a patient's tumor and then look at the expression of a vast amount of genes, and then compare that to what is normally expressed in that cell type. So if we're looking at, for example, a lung tumor, then we can look at what's normally expressed in those lung cells, and then see what genes are turned on too much and overexpressed and what genes are turned off. So right here is a picture of a cell, a tumor cell. And what you see on the outside of these tumor cells is you see a bunch of receptors. Now, these are things that poke out of the cell surface, and they're usually used to attach to molecules that are being taken into the cell. There's also a bunch of molecules that are used by the cell to basically like a street address to tell the immune system what this cell is and what the contents of the cell are. They're referred to as antigens. And an antigen is anything that causes the body to make an antibody. Antigen stands for antibody generator. And so our cell surfaces are littered with these different proteins that can be used to characterize a cell. And most importantly, tumor cells have unique levels of these proteins, and they often have higher levels of certain cell surface receptors or antigens that are not found in normal cells. And so this technology has really changed the way we treat cancer. There's a class of drugs called antibody drug conjugates. And what these are antibodies against a certain cell receptor or antigen that is commonly found in tumor cells and not so much found in normal cells. And what they do is they have this antibody, they create an antibody. It's an easy process. You can create an antibody for just about anything these days. And they create an antibody against a certain cell receptor that is highly overexpressed in certain types of cancers. And then they attach a molecule of a very potent chemotherapy drug. So over the years, there's been many different chemotherapy drugs developed. Some of them are just too toxic to be used properly in a systemic level. In other words, when they inject them into you or you take them orally, they kill too many normal cells. This process of antibody drug conjugates is like a targeted chemotherapy. It delivers one molecule or a few molecules of that chemotherapy compound directly to the cell, and it attaches it to the cell. So you have much less chance of side effects, and you also have the ability to seek out and find tumor cells anywhere in the body. And so the first antibody drug conjugates were designed a few years ago. Since then, many companies have jumped on board and they've realized, wow, this is the way to do it. And so as you can see there in this article by Huang et al., 2020, they have this cell here, and you can see those receptors, and you can see the antibody drug conjugate is targeting that receptor, and those little dots there, those little red dots are the chemotherapy agents. And they're going to get taken into the chemo cell or the tumor cell, and the chemo's going to be released and it's going to kill the cell. So we're going to go into the next slide now. So as of November 2020, FDA has approved 14 different ADCs. Now, when I look at the different clinical trials, there's a lot of companies building ADCs now against different proteins and cell surface receptors. So this is really the way to go. So here's a little short list, just some of them. And so you can see the drug name. They've always got two different names. The first name is the antibody that targets that cell receptor or antigen, and then the second word stands for the chemotherapy name. Now, I don't know where they come up with these names, but they're very interesting names. Infortumab, anything with MAB stands for monoclonal antibody. So if you see MAB on the end of a word, it means monoclonal antibody. And then if you look at the target, you'll see actually what it targets. So you can see, for example, N4-tumab vedotin targets Nectin-4, sacituzumab govitecan targets the TROP2 receptor, lots of different receptors here. So I'm just going to go on to what we do. So the way we identify what receptors are overexpressed in a person's cancer, we isolate their tumor cells using a new technology called laser dissection. This is really important. For RNA expression testing, you want to make sure you're only using tumor cells. So as most people know, especially if you have a pathology background, that most tumor biopsies are maybe 20 to 40% tumor cells. The rest are a variety of different body cells and immune cells and fibroblasts and all kinds of different cells. We want to know what the tumor is expressing. And so this laser dissection is a great new tool that can be used to isolate a purified sample of tumor cells. Now, it's a fairly rigorous process. We had to have an expert pathologist in the States, and this is all they do. They isolate tumor cells from biopsies. And so what they do is they use this laser beam to isolate the tumor cells, and they get a purified vial of the tumor RNA. And then they extract the RNA and they ship it to us. And so we have a next generation sequencing panel, and it looks at 20,813 genes. That's a lot of genes. And it counts the number of RNA molecules and reports them as reads per million. We then compare that to we have created actually a database of different normal tissue expressions. So, everything from glial cells in the brain to liver cells, all kinds of different cervical cells, cervical squamous cells. We have this huge database that's taken us quite a while to create of normal expression. And we use the exact same techniques and the same tools to create those normal cells. And we basically overlay them. And we have a computer model that basically compares the two and comes up with a fold change. In other words, how much over or under expressed is a certain gene. So the next slide, I'm going to give you an example of that. And so here's a little snippet of a certain case we had. In this particular case, you see what the tumor RPM expression, reads per million expression, is on the far left. Then you have the gene name, and then you have the normal version of, the normal lymph node tissues. So in this particular case, we're looking at lymph node metastasis in this patient. We wanted to know what the normal expression was. And so you'll see certain genes like ALK. So ALK is an interesting gene. It's only expressed embryonically. And sometimes tumors will turn it on. And if it's turned on, then we know it's a cancer that's creating it. And so there are probably about 7 different drugs that are approved to target ALK. In this particular case, the patient's tumor was not using ALK, as you can see. And so there was 0 found in the tumor and then also 0 in the lymph node, which is expected. So there was no fold change there. And then we can look at certain genes like, for example, ATM. That's a tumor suppressor gene. So that's normally turned on. And it's supposed to be turned on all the time. And it can definitely help prevent certain, for example, it's involved in colorectal cancers. It's turned off in colorectal cancers. And in this particular case, this was a colorectal cancer. And as you can see, the tumor RPM is quite low compared to the normal lymph node. And so this gene was actually underexpressed by minus 6.4 fold. And so obviously that's one of the causes here. So if we look at AKT1, we can see that's slightly overexpressed, 1.3. In order for us to, say that it's really overexpressed, we want to see at least a, 4 or 5-fold overexpression. The higher the overexpression, the more the tumor is using that as a way of metastasizing and growing, and the more impacted the tumor will be if we inhibit it with a targeted drug that inhibits that. So I'll give you a couple of case examples here. So recently we had a case example of a patient that was offered a clinical trial. So the patient said, well, my doctor's, suggesting this new trial. She didn't really know anything about the trial. She just said it's this new drug called anatumumab ravtansine. And she said that some patients apparently were getting a good response and others weren't. And we had no clue whether it's going to work in that patient. So what we did is we did RNA expression testing on their tumor. And we found, first off, what we did is we looked up the drug, of course, and we found that the drug targeted a protein on the outside of a cell called mesothelin, and that's known as MSLN. And so this drug, basically, anatumumab is an antibody that targets mesothelin on the outside of a cell, and then it attaches a chemotherapy drug to that. And so we want to know if this patient's tumors expressed MSLN, the mesothelin gene. If they did, then this drug would offer a great tool for them. So we found out that it was actually overexpressed by 53.92-fold in the patient's tumor sample. And we encouraged them to enter the trial. And so the patient's been on the trial for a short period of time, but has had a phenomenal response. So here's a great example of this, helping find new therapies. Oops, sorry. And then, so the next slide. So here's another case example. In this particular case, the patient's tumor DNA sequencing showed that they had many copies of EGFR. EGFR stands for epidermal growth factor receptor. It's a gene that has quite a few different targeted therapies, has monoclonal antibodies against it, has tyrosine kinase inhibitors. In this particular case, it was a lung cancer. And although the patient didn't have an EGFR mutation, there was increased copies of EGFR. And so this can be an FDA-approved treatment for people if they have, EGFR inhibitors can work very well in certain lung cancers. And so the doctor actually asked us, well, the patient's got increased copies of EGFR, and usually when you have increased gene copies, that leads to overexpression in a lot of cases. However, not always. Sometimes those genes can be copied in a faulty way. So in other words, the genes are copied by the cancer. It's part of its standard process. Many genes are copied, in cancers. It's part of the chromosomal aberrations that happen. And, often you get these increased oncogenes. So EGFR is considered an oncogene. It causes cells to grow and metastasize when it's overexpressed or mutated in a certain way. And so the doctor said, well, I want to put the patient on this drug. They've got multiple copies. And so I said, well, let's make sure that they actually do have expression of it, because there's no point putting the patient on the drug if they don't have high EGFR expression. Just because there's multiple copies doesn't necessarily mean that the gene has been copied in a functional manner and that there's a functional version of the gene being used by the tumor. So, should the patient go on an EGFR inhibitor or not? These drugs do have some side effects. And of course, you don't want to waste time on a drug that's not going to work for you. So we looked at the RNA expression, and we found that EGFR was not overexpressed. And so obviously, in this particular case, the multiple copies were non-functioning copies. And you wouldn't be able to tell that if you just went by the DNA. But since we have this wonderful tool, we're able to say, hey, this isn't a good drug for the patient, but there are other options. So, I'm going to give you an example. One last screenshot here. Oops. So I did a quick search of, what drugs, how many antibody drug conjugates. I just did a Google search in clinicaltrials.gov, which is the international clinical trial database. And, using a search like antibody drug conjugates is not the best. There's probably a lot more of these because many of these trials don't list the drug. They list the drug name, but they don't tell you what it is. They don't tell you that it's an antibody drug conjugate. So this is just looking for clinical trials with the term antibody drug conjugate. There's probably 3 or 4 times more of these drugs actually in clinical trials than this, but there's 229 studies of antibody drug conjugates. So, here's a great example of how many different treatment options there may be for a cancer patient. And the beauty of these drug conjugates is that they work across many different tumor types. So if you have a tumor that is overexpressing the tropic receptor, doesn't really matter what type of tumor you have. It's going to target this drug, sacituzumab govitecan, is going to target it regardless of the type of cancer you have. So it's very different from saying, well, this drug is approved for breast cancer, but it won't work in lung cancer. These drug conjugates are very different. They can be applied across a bunch of different types of cancers. And it's all based on whether you have expression of that receptor or not. So this tool is changing everything for us now. We can determine whether certain genetic alterations like copy number variations found in tumor DNA sequencing is actually targetable or not. And we can identify targets and drugs that tumor DNA sequencing just wouldn't find.
I'm sure there's a lot more we could go over, but I think that's enough for now. DNA is not enough is the fundamental point here.
DNA is not enough. That should be a song.
Yeah, DNA is not enough. RNA testing is here. It is accessible. It is affordable. And again, it's going to make sure that your doctor dials in the perfect treatment for you, as opposed to what everyone else gets based on, on what type of cancer they have rather than your mutations. And we know cancer is a disease of the genes. You can't have cancer without having mutations. So if you've been don't have markers or you don't have any, that is absolutely how you know your doctor did not cast a very wide net, and you need to take matters into your own hands. That's my public service announcement. Okay, thanks, Alex.
Thank you.